Welcome to Chapter 14 of Anatomy Physiology, and we're going to be talking about your brain. So I love optical illusions, and so I thought I'd throw one in here. You can't see all the little black dots. Your brain can't focus on all of them at the same time, but they're there. And then I wanted to tell you this. Whether you think you can or you think you can't, you are right. So if you tell yourself you can't do something, then you're correct. You can't do it. But if you tell yourself you can do things, most things are possible. So there's your little pep talk. And let's get into this. So we're going to try to take a whole course of neuroanatomy and cram it into 1 or 2 hours. So obviously, we're just going to touch bases with some of the big concepts, and then you feel free to go and learn a lot more, because the brain is endlessly fascinating. And then of course you can get off on psychology and find out how people usually act, and there's futuristic stuff, so I'll throw you futuristic stuff in there too that we probably will have very soon. I'll start out with one of the concepts that's kind of an interesting one. So they talk about being brain dead, or the person is not alive anymore. Even though their heart's still beating, they're brain dead. There's no conscious thought going on, and that the person cannot wake back up. So usually they talk about pulling the plug and donating the organs to someone who can use them. But here's an interesting thought. They've started transplanting hearts from people, and if you remember from the chapters on muscles, you have nerves, you have electricity, you have innervation from the nerves in the heart. Although the heart has its own pacemaker, it can respond to other nerves. But they've transplanted hearts from one person to another, and the person who receives the heart a lot of times knows things about the recipient that they couldn't possibly know, because they don't tell you whose heart you got. That's one of the rules, that you don't know. So you don't know whether it was from a child, from an older person, from a boy, from a girl, you don't know. But yet, case after case, if you go and look online, you find people who got a heart and suddenly know things about the person who donated the heart. So wouldn't it be interesting if some of our memories are stored in the heart as well as in the brain? I put this in as something you might want to look at. There's a man that was dying, and he felt badly. He was in jail, and he said, well, let me donate my body to science so that, that might help from a lot of the bad things that I've done. So he gave it to this plastination company, and they took the man and they turned him into plastic. They preserved him in plastic. And then they sliced him so that you can see every few millimeters of this man, looking all the way through him. And then there was a woman said, well, I'm dying and I would like to do that too. And so she donated her body. And so we had the plastinated man and we had the plastinated woman. And now this is a company, and you can buy plastinated specimens of people who've passed on. You could also, they have a traveling show, so you can actually go see it. So I've actually gone to see the show. If you look at earlier man, and then you look at more modern Homo sapiens, you're going to find a lot of changes, especially in the brain. The spinal cord pretty much has stayed the same, but we have better vision now than they used to have. Our memory is greater, and our control of our prehensile hand. So if you take your thumb and you cross it across your palm, that's prehensile. So we have the ability to do that. We have opposable thumbs, and we can grasp where a lot of things they don't have prehensile, so they can't grasp like that. When you look at the brain and you slice it open, look around inside of it, you find some of it looks gray, some of it looks white, and then there's some black spots here and there. So we're going to find out what the colors of the brain mean. Coming out of the brain are 12 nerves. So remember, those are bundles of axons. Most of them are going to be able to send and receive, so they have afferent and efferent paths in them, but 3 of them are sensory. So one of them is the olfactory, a sense of smell, and your optic nerve, because you see with your optic nerve. The number 8 nerve is for hearing. So those are your 3 sensory ones. The 12 cranial nerves are numbered with Roman numerals, and Roman numeral 1 is your olfactory nerve, the one you smell with. So we have an olfactory bulb. Here's the frontal region of the brain. And then you have the cribriform plate. We learned about that when we studied the skull. And it has little holes in it that allow these nerves to come down into the nose, and they sample the nasal mucosa, the snot, basically, that's in your nose. And it sends the information up through the olfactory bulb. The number 2 is the optic nerve, and you have one coming from each eye. It comes together at the optic chiasm, right there, and then it goes back to the occipital lobe, which you can't see here, to understand what the eye is seeing. So your eyes can work just fine, but if the occipital lobe isn't working, you don't know what it is you're seeing. So it's just light coming in. Always remember your pituitary hangs down the optic chiasm, and right above it is your hypothalamus. And there's a little stalk that dangles down in between your optic nerves that allows the pituitary to sit down in the sella, S-E-L-L-A, the sella of your skull. So there's a little bony pocket just for the pituitary. And that's going to be important when we talk about the endocrine system. The oculomotor nerve, number 3, is the one that allows your eye to move up, forward, and down. So it allows you to look up and down. So, and it tells you what it does. Oculo means eye, motor means move. So this is the nerve that moves your eye. This is Roman numeral 4, and this is your trochlear nerve. It innervates the superior oblique muscle. So when you learn the muscles of the eye in chapter 16, this is one of the things we're going to learn about, these different muscles that pull the eye in different directions so that you can move your eye freely. But you have one nerve just dedicated to one muscle, so that's kind of unusual. The 5th of the 12 cranial nerves is one of the more interesting of all of them. First of all, it has 3 origins, so it branches off 3 different ways, and it covers pretty much from your forehead, above your lips, beside your nose, down to your chin. Now, what's interesting is they've tracked people who have migraines to trigeminal nerve problems. And a lot of times before someone has a migraine attack, they'll get a tingling in their chin or a tingling in this area around their eye. So that's kind of an interesting thing right there. Another thing that's kind of interesting is if you pinch this off, so we've talked about in the carpals of your hand, you have the retinaculum, you have a tight band of connective tissue that holds very tightly. And if you move your fingers and you're sliding those tendons back and forth, then you can get irritation. and we call it carpal tunnel syndrome. So you can get something that's a little bit similar to that, because this has to pass through a little passageway to come out into your face. This is an interesting map showing where the different divisions innervate. So they've kind of lit up the 3 different regions. The top branch, the ophthalmic branch, is sensory, and then the maxillary is also sensory, but the mandibular sends signals both directions. So that's why they call it a mixed nerve, because the axons can carry messages to and from. And I put this in because it's partly funny and partly true. So if you get a migraine, you're so sensitive to light, you just have to go into a dark, quiet place until it passes. If you get a headache from having high blood pressure, if you have stress headaches, and if you're out Christmas shopping. So here's the Bell's palsy, where you have inflamed the nerve. But there's one called tic douloureux, and this one is intense stabbing pain. So just touching your face, trying to drink something, even running a washcloth over your face causes you to just scream in pain. And it said that the treatment may actually require cutting the nerve. If it doesn't resolve, then the pain is so intense that, obviously you have to be able to drink. You have to be able to put food in your mouth. So you can't live very long with this if you can't control it. It's unfortunate they put both of these cranial nerve disorders on one slide, because they both have to do with the face. But the trigeminal neuralgia, the tic douloureux, is with the trigeminal nerve, which is the number 5 nerve, and Bell's palsy is the facial nerve, which is the number 7 nerve. In Bell's palsy, your mouth is going to sag down so you can't smile. You're going to be drooling quite a bit, and you have trouble closing your eyes, so your eyes tend to dry out. So you have to keep putting fluid on your eyes. It usually resolves itself within 6 months, but older people sometimes get it and it never resolves itself. So they end up still having partial paralysis of their face. The way I remember the abducens nerve, it's your number 6 nerve of your cranial nerves. It pulls the lateral rectus muscle, so you are able to look over to the side. So when you abduct someone, so abducens nerve, when you abduct somebody, you look out of the corner of your eye to see if anybody's checking and seeing that you're about to snatch a kid. So that's how I put it together. You have to have some memory device to put these things in your head, and that's just the one that I came up with. So that's your abducens nerve, number 6. And here's the number 7 facial nerve, and this is the one, it looks a little bit like the trigeminal coming out, but it branches off and goes a little bit deeper. So you're going down into the salivation, the tear glands. If you remember, the trigeminal came all the way up into the forehead and came all the way down into the mandibular region. And the facial nerve, number 7, doesn't reach that far. The facial nerve is in charge of the front, the anterior 2/3 of your tongue, and it's your sensory. So if you have damage, you're not going to be able to taste sweet and salty. The branches are the temporal, so it goes up to your temple. Zygomatic, so that's your cheekbone. Buccal is your cheek. And mandibular, your mandible and your maxilla. Remember that you keep those two apart. and the cervical region. And this is not the cervix in the vaginal region. This is the cervix, which is the top of your spinal column. I thought this was a really clever way of remembering where the facial nerve branches off. Right there. I'm not ready to get it tattooed on my hand just yet. And if you remember, the number 8 nerve is the third sensory nerve, and it's the vestibulocochlear. So inside the ear you have the cochlea, and look, there is that nerve. And then here's your semicircular canals, which we'll discuss in chapter 16, that allows you to balance. And there is the vestibule of the semicircular canal, and that's why they call it the vestibulocochlear nerve. When you see the word glosso, then you know that you're talking about the tongue, and the pharynx is the back of your mouth, so you go all the way back to the back of your tongue, and then that opening there where you can go down into the esophagus or down into the airway or up into the eye or up into the nose. That's the pharynx, or back of your mouth, or the top of your throat. So the glossopharyngeal nerve innervates that area. You have salivary glands at the back of your tongue, and that's some of the ones that are innervated. The vagus nerve innervates your smooth muscles. So this is an extremely important, the number 10 nerve. So this is going to innervate or synapse with your hollow organs, because most of your hollow organs have smooth muscle. Your stomach, your esophagus, your intestines. It has the most extensive distribution of any cranial nerve. So you had one, like the trochlea, that just had one muscle that it innervated. But this one has a major role in controlling your heart, your lungs, your digestion, and your urinary. Your ability to swallow, your ability to talk, and the regulation of your viscera, which is mostly your intestines and hollow organs that are inside your abdomen. If you have damage to the vagus nerve, you're going to either be hoarse or lose your voice. You're going to have trouble swallowing. And if you damage the vagus nerve, it can be fatal. In these pictures, it shows you one of the nerves, but you have to remember that you have 2 hemispheres of the brain. You have 2 sets of these cranial nerves. So just keep that in mind. Now the accessory nerves are some of the weirdest looking ones. There, all of that is the accessory nerve coming down there. And then there's the other one over there. So it's rather odd looking. The interesting thing that these do is they innervate the sternocleidomastoid muscle, and that's going to help you turn your head, and the trapezius muscle, which helps you shrug, or as I tell my students, I call it turtling, where you pull your head down and you pull your shoulders up like a turtle does. Another interesting thing is these are some of the crossover muscles, or nerves. And so the one on one side talks to the muscle on the opposite side, the contralateral side. So these are crossing over. And the 12th of the cranial nerves is the hypoglossal nerve, number 12, and it is below the tongue. The number 1 nerve is your olfactory, and then 4 of your others are involved with the eyeball. So the optic is number 2, the oculomotor is number 3, trochlear is number 4, and abducens is number 6. Trigeminal is number 5. Facial is number 7. Vestibulocochlear is number 8. Then you have your tongue, throat nerve, glossopharyngeal, which is the number 9. Number 10 is going to be your vagus nerve. And remember, it is the farthest reaching. It has its little nerve endings in most of the things going on in your body. So it's super, super important. And then your number 11 nerve, your accessory nerve, is for shrugging and tossing your head. So you remember that one by going, whatever. Because usually you do that by jerking your head from side to side and bringing your shoulders up, going, whatever. And then last but not least is number 12, which is your below the tongue nerve, or your hypoglossal. So now we spent time going over the 12 cranial nerves. Now you come down to the spinal column, and you have the nerves that branch off from the spinal column, and they're threaded through the vertebra for protection and support. And then they branch off into other nerves, and this is one I just thought I would throw this in. The ulnar nerve, we're seeing kids holding their cell phone for such a long period of time that they actually pinch the ulnar nerve and they cut off blood flow. So then you end up numbing your ring finger and your little finger because of the ulnar nerve being put to sleep. Another thing that you can do with the ulnar nerve is if you put your elbow down too quickly, you can pinch it, and they call it hitting your funny bone. And it's not hitting the funny bone, you're actually hitting the ulnar nerve. This is an interesting picture. It shows you different ways you can see different things in the brain. So you can just do an x-ray. You can inject a contrast dye and look and see where the blood flows and if there's any constrictions. Then you can use a CT scan. This is what a CT scan looks like. They're less expensive, and they don't quite give you the clarity that you get if you do an MRI. So here's an MRI. And then the PET scan, the positron emission, this one, you actually inject the person with radioactive glucose, and you look to see where in the brain it's being used. What area of the brain is it pulling it in? So this one's kind of an interesting. And then, of course, I have to throw in a joke. And here is Monty Python quotes. This is where you store those in your brain. Here's where all the comic books you read, useless trivia, cartoon theme songs, obscure science fiction references, internet memes, and a little bit of science and math. Of course, this is a joke. This is not where we store this information. But we did learn to use the PET scans to look and see if you say something, then the visual cortex lights up here. So they said the word car and that lit up. If you are thinking about a verb, drive, an action, the Wernicke area will light up. And if you try to speak, then you have an area, the Broca's area, Broca's brain. And then the primary motor cortex. So you are executing the whole thing. You have car, drive, and you want to say drive. So you need to be able to speak it. And so you've integrated the whole thing. So there's your primary motor cortex. So we had been able to map out the parts of the brain that were involved with the various functions. And that was pretty exciting. We also were able to do PET scans of normal people versus people who have Alzheimer's. And you can see that a lot of the areas of the brain are no longer functioning in a person who has Alzheimer's. So they've actually destroyed these areas by putting amyloid plaques in there. proteins that clog up and kill the nerves. And then we develop the MRI, and it can show us so much more than the PET scans. So the one thing that's a downside of giving an MRI is they put you in a tube. Now they've gotten better over time, but if you're claustrophobic, it will scare you to death to stick you inside of an MRI tube. So here they are wanting to see why this woman has claustrophobia. So they're about to stick her in this tube, and they're going to find out that she does indeed have claustrophobia. So she probably won't be able to have the MRI. You can also see multiple sclerosis with an MRI. So now they've developed functional magnetic resonance imaging, or fMRI. And I've put a link down here. You can go look at 60 Minutes mind reading, or you can just click on this link, or type in this link and get there. But Leslie Stahl did a fascinating report, and she's talking with a guy who does fMRI, and he can put you in an MRI machine, and he can tell you what you're thinking. He can exactly tell you that you're thinking about a hammer or a car, things like that. And through there, they tell you other things, like if you've been in a room before, and you say, no, I've never been in that room before, they can put you in the MRI machine and they can show you pictures of the room, and they can see your brain light up and your brain remembering being in that room. So they're thinking that this is so much better than a lie detector, because you can actually see that the person is lying and that they've never, they have been in the room before. So this particular episode of 60 Minutes goes through different things that they can do with functional MRI to do mind reading. And it is really awesome and scary at what they can do and what they can read in your mind. So here are some terms that you need to memorize, and one of them is, if you're going towards the forehead, they call it rostral, and if you're going down towards the spinal cord, it's caudal. And you need to know that there's a longitudinal fissure that separates the two halves of your brain. So you truly have a left brain and a right brain. Here's that picture of someone who has multiple sclerosis, but you can see here's one hemisphere over here, and here's one hemisphere over here. So you can see that there's two halves to the brain. If you're looking at the brain, you have thick folds that stick up, and they call those gyri. So one fold would be a gyrus, and this is gyri, or all of them. And you have little grooves or valleys in between them, and they call them sulci or sulcus if there's just one. And so I remember that because when you're sulking, you pull your mouth down, and when you're in a sulcus, you're down in a groove. And then you have the corpus callosum that connects the two halves of the brain. So they can think independently of each other, but if you want one side to talk to the other, you kind of have to send it through this, almost like an internet connection, the corpus callosum. So here's a picture. Here you have one half, here you have the other half. There you have the longitudinal cerebral fissure. So a fissure is much deeper than a sulcus. A sulcus is just the kind of a groove in between there. So this is a sulcus, there's a sulcus right there. And then this thing sticking up, this bulge up, that's the gyrus. So there's your gyri and your sulci, and there's a fissure. And you have a couple of other important fissures that you're expected to know in lab. There's your frontal lobe, your parietal lobe, your occipital lobe. And if you could come here over on the side, then you would see your temporal lobe over your temples. The central sulcus is important because it's kind of midway, but it comes down and intersects with the longitudinal cerebral fissure. The majority of your brain is the cerebrum, or the thinking part, and that's the part that has the left and the right hemisphere. And they talk about people being right-brained and left-brained. It's made up of the gyri and the sulci and the longitudinal cerebral fissure and the corpus callosum. And then you have the cerebellum, which is a kind of a wadded up underneath the cerebrum. So there it is. It's completely different. Doesn't have the sulci or the gyri. And it's involved with balance. And then you have your brainstem, which is the next part, and then you go down into your spinal cord. And this is similar to the picture that you should have had in lab. And the landmark that I use is the corpus callosum, because it is so obvious. When you cut the brain in half, you can see that right there. And this extends back into this hemisphere and over into the other hemisphere that we've cut away so that you can look inside. When you come down from there, you come to the thalamus, which is this area. And then under the thalamus is the hypothalamus. And then under that, you have the optic chiasm. So that's where your two optic nerves are. And then hanging down beneath that is your pituitary. So the four things are kind of stacked. The thalamus, the hypothalamus, the optic chiasm, and then the pituitary hanging down off the tuberoinfundibular stalk. But you don't have to know that. All right, and then when you cut into the cerebellum, you see something that looks like a tree, the tree of life. And so they call that the arbor vitae, the tree of life. If people have whole brain epileptic seizures to the point where it's life-threatening or it's so destructive that the person can't even have a remotely normal life, they, in extreme circumstances, will cut the corpus callosum. So then if you have an epileptic seizure, it will be confined to one hemisphere and it can't pass through the corpus callosum over to the other side. But that's very radical. They don't do that unless it's life-threatening. So the other picture was a cartoon or an artist drawing of what the brain looks like, but this is an actual brain where they've cut it open, and you can see the pons, and you can see the medulla oblongata, tree of life, arbor vitae of the cerebellum, and then the whole cerebrum there. And as promised, I would tell you a little bit about the color. So if you're looking at the gray matter of the brain, that's where your cell bodies are. That's where your soma, your neurosoma are. That's the part that has the nucleus, the mitochondria, all of that stuff. There's very little myelin. So myelin is just basically fat. It's phospholipid bilayers, and it's white. So just think about Crisco covering your muscles, or excuse me, your nerves. And then your axons have the myelin. So we call it the white matter because it is white. Now, if I were designing the brain, if I were a superior being, it seems odd to me that I would put the most delicate and easy to kill part on the outside. But the outside is where the gray matter is. That's where your cell bodies are and your dendrites. And then deep inside, underneath the gray matter, that's where you're going to have the axons. So I just find that odd. Now you can regenerate an axon, but you can't regenerate a cell body. If you destroy the cell body, if you kill the nucleus, then the cell has no way of coming back from that. But you can regrow an axon. So anyway, I just always find that odd. There's a section in the chapter where it talks about embryonic development, and I'm not going to talk about that except for you should know that when you're making the neural tube, you make a neural groove and then it curls up into a tube, a hollow neural tube. If the mother doesn't have enough folic acid, oddly enough, then this may not finish closing up. And so the child will have what we call spina bifida. It can be life-threatening because you can't hold all the spinal fluid in that you need to, the cerebrospinal fluid. Sometimes it's such a small leak that it actually doesn't cause problems. Sometimes you don't even know it until they do an x-ray or something and they happen to see, oh, by the way, did you know you have spina bifida? It's also interesting to note how developed the brain is by the 4th week of pregnancy. Usually the mother has just discovered that she's pregnant. By all means, read over it and see how the brain develops. But again, that's out of the purview of this course. That's beyond the scope of this course. I used to know the man who laid out the bodies at one of the nearby universities for the medical students, and so I asked him if I could bring my students in and we could look at some of the bodies, and he laid one out for us and went through it, and it was absolutely fascinating. The kids were at first a little leery about seeing a dead person, and then they were so excited and they were taking pictures, and I was just like they were taking pictures. So here's one I took of the brain, and you can see the meninges of the brain covering it. And then as you see where it has been torn away so that the students can look at the brain, you can see the sulci and the gyri. So there's a gyrus right there, and there's a sulcus. And then here you're coming out. This is the brain stem. You're coming out, and there's your spinal cord. It's absolutely fascinating. Another thing that surprised me when I was looking at it was that the brain, I just assumed that the skull would all be the same thickness. And when you look at it, when they've cut it open so that you can take the brain out and look at it, you can see that some places are very thin and other places are thicker. The meninges are 3 membranes, and they make a continuous cover over the brain and over the spinal cord. And they hold in the cerebrospinal fluid. They protect the brain. They are the structural framework for the arteries and the veins. And the outer layer is the tough mother, the dura mater, the tough mother. And then the innermost is the loving mother. And so it nestles up against the brain and holds it. It goes down in the sulci and up over the gyri and then down the spinal column and actually touches it. So it's the loving mother. And then in between them is the arachnoid mater. So the spider mother. Here's another picture where they've cut away the skull. So you can see the hair, the scalp, the skull. And then there's the dura mater right there. And when you peel it back, the arachnoid sticks with it. So they go together. But the pia mater often is left behind. So the pia mater is still laying on top of this area right here. And you can see the blood vessels within the dura mater, underneath the dura mater. It's between the arachnoid and the pia mater is where you're going to find most of these blood vessels running through there because there's a space there. If you get a brain bleed, sometimes you hear about a brain bleed, you get a subdural hematoma. So you're bleeding underneath, and it can be life-threatening because the tough mother, the dura mater, does not give way. And of course, the skull does not give way. So any pressure that builds up from the bleeding from wherever you've damaged a blood vessel, it's going to be pushing down on the brain. because it can't push out through the dura mater. So sometimes they actually have to drill a hole and relieve the pressure because you got too much pressure building up. Another thing that can cause pressure to build up would be if you get an inflammation of the meninges, and that would be meningitis. And one of the things that whenever I would have like a blinding headache, I was like, oh my gosh, am I dying? Am I dying? Do I have encephalitis? Do I have meningitis? And you can check yourself. If you have so much swelling in here from the meningitis, you won't be able to pull your head down onto your chest because there's too much fluid in the cranial cavity and then down the spinal cord to allow you to do that. So meningitis can be a scary thing because it can crush parts of the brain. So you can end up deaf. So there's a lot of, or dead. We now have a vaccine for meningitis, so a lot of children are now getting that. So we should see a lot less deaths from meningitis in the future, or a lot less deafness or other brain damage from survivors. In the skull, the meninges, actually the tough mother, the dura mater, pushes up against the cranial bone. So it's actually up against the skull. And so you don't have a space there. But now when we get into the spinal cord, the spinal column, you're actually going to see that outside of the dura mater, you have a space. And we call it the epidural space because it is upon the dural space. So it's outside. Now, only the spinal cord has it. The brain doesn't have this. But for ladies who are having childbirth, the epidural space is a wonderful space because you can put a little numbing agent outside the spinal cord in the epidural space, and it will numb you from there down. So you can deliver your child without experience the little pressure that the doctor says you'll feel when the baby comes through. I always think, since I've had children, and they say, you'll just feel a little pressure when the baby comes out. And I have to bite my tongue not to say all kinds of things about that. Because I was before they had epidurals when I had my children. So I didn't have that benefit. The pia mater is so thin, you really need a microscope to be able to see it. So there's your pia mater right there. And then if you notice the arachnoid, it's got these little spacing structures that allow fluid and allow the blood vessels. So that's what we were seeing before. And then, of course, the tough mother is right up against the bone. When you take Med Micro, one of the things you will look at is this particular bacteria that causes meningitis. If it hits a child when they're 3 months to 2 years of age, a lot of times it can cause death. Now, you can have bacterial meningitis and you can have viral meningitis. Itis just means inflammation of. So you have inflamed the meninges with something. But they have the bacterial vaccine now. For those of you who want to follow up on this, I found a couple of sites. You can look at meningococcal vaccination. So a coccus is a bacteria that's like a sphere. That's a coccus. So there are 2 types of vaccines available. So here's information from the CDC, here's information from Harvard, and here's information, again, from the CDC. And here is some information. You have the stiff neck. So meningitis can also be caused by a fungus. You lose your appetite, you run a fever, you're sleepy, you're lethargic, and you have a headache. So you can recover within 2 weeks, but the bacterial form is often life-threatening. So you need to treat those. The viral kind, we always think of viruses as being the scary ones, but if you get a viral meningitis, usually it resolves itself. So it's much less dangerous than the ones that are caused by the coccus bacteria. In order to diagnose it, you have to do a lumbar puncture. So you get some of the cerebrospinal fluid that's bathing the brain and bathing the spinal cord. So it's called a spinal tap. But it can kill you very quickly. You can go into a coma and then die within hours of getting it. So speaking of cerebrospinal fluid, you also have inside your brain 4 fluid-filled chambers, and they call them ventricles. So you have ventricles in your heart, and those are blood-filled, but in your brain, the ventricles are fluid-filled, and that's cerebrospinal fluid, or CSF for short. And here's a picture showing it's very easy to see where the holes are in your brain. So when somebody says you have holes in your brain, you say, well, yes, I do. As a matter of fact, they're called ventricles. And they have cerebrospinal fluid that flows through it, and there's actually little cells that are waving it through. So, but this is what it looks like. It almost kind of looks like something out of Star Trek. So there's the caudal view, the rostral view, so a side view right there. There's a ventricle right there. Here is the anterior view. So you're looking, so your eyes would be up here somewhere on this side, and then you're looking back. So there's your frontal lobe, and then you can tell because your cerebellum is underneath the cerebrum towards the back. It's posterior. So there is another view of the ventricles there. I like this picture because it shows you exactly where they cut to get this view. So this is a cartoon picture, but this is an actual slice out of a human's brain. And you can see the corpus callosum connecting the 2 hemispheres there. And you can see that deep, deep fissure that divides the 2 hemispheres. There's your gray matter on the cortex or the outside, and then there's your white matter on the inside. So those are axons, and those are the cell bodies. We talked about neuroglia in another chapter, and we're going to revisit it. So there's different kinds of neuroglia, and one of them is the ependymal cells, and these are the ones, this is the neuroglia, that lines the ventricles, and its function is to modify the cerebrospinal fluid. So it has more sodium, more chlorine, and less potassium than you would find in plasma. So there's a very little protein in the cerebrospinal fluid. There's quite a bit of glucose because you do need glucose to keep the brain happy, but you don't have as much as you do in the plasma. So you have less potassium, less calcium, less glucose, and very little protein, but a lot of salt. Sodium and chlorine. Sodium chloride is table salt. The ependymal cells have cilia, and they push the fluid. So it's continuously bathing the brain and bathing the spinal column. I find it interesting that they put the flow of the cerebrospinal fluid in an introductory anatomy physiology textbook because this is way, way more than you guys need to know. You definitely want to know the functions of this CSF though. So it allows the brain to float. So it's buoyant. It's actually floating in the cerebrospinal fluid. So it isn't crushed. The brain is actually so very soft. If you've ever held a brain in your hand, hopefully not human brain, but if you have ever felt a brain, it's very, the spongy, more gelatin feeling. So if your brain was sitting on the floor of the skull, of the cranium, the pressure would kill the nervous tissue. So if for some reason you lose cerebrospinal fluid, for example, they do a spinal tap and take too much fluid out, or if you have spina bifida and the end starts leaking, you may have not enough fluid to float the brain, and so it comes down and it starts killing the bottom of the brain, the cerebrum. And of course, you've also got the brain stem coming down through the foramen magnum. So you need to have it up. You can imagine if you've fallen asleep on your arm funny and you wake up and your arm is asleep because you put pressure on it and the blood flow couldn't get through it. So you can kind of imagine what would happen if the brain was sitting down on that hard skull, the bone of the skull. Another thing it does is it slows down the brain. So if you've ever tried to run through water, that it impedes your progress. You're trying to run through the water and you just can't go as fast. So if your brain is having to move through fluid, through the cerebrospinal fluid, then it's not as likely to hurt when you hit against the bone, when it goes flying forward and hits the bone. So if you're in a wreck, if you're a boxer and somebody hits you in the head, or heaven forbid, shaken child syndrome. Why a parent would think picking your child up who's crying and shaking it really hard would make it stop crying, I've never figured that out, because that just makes them cry harder, but it also can kill them. So a lot of times they'll bring a baby in and say, oh, they died of they say, oh, my child had SIDS, sudden infant death syndrome, or also called crib death. It's extremely rare. There's fewer than 1,000 cases per year. It usually is going to happen. It happens in a boy, and it's usually before the age of 1. So I remember being told that with my children, and so pretty much I'd listen all night long to see if my child stopped breathing, because I was so terrified. Had I known that it was less than 1,000 cases a year, I probably wouldn't have worried quite as much. But a lot of people who shake their baby to death will take it in and say, oh, it's sudden infant death, syndrome. It's SIDS. They just died in their crib, and we don't know why. And then during the autopsy, they see the bruising, and they realize, no, that's actually not how the child died. And similar to the way that plasma carries away waste so that we can excrete it through our urine, the CSF is going to wash away metabolic waste from the nervous tissue. So any exocytosis of waste from the neurons and the neuroglia can combine with the CSF, and it can be rinsed away so that you can filter it out. Now I want to spend a little time talking about the blood-brain barrier and the blood supply to the brain, and give you a really interesting fact that a lot of people don't know, but your brain doesn't feel pain. It understands pain. It processes information from the rest of the body saying, ow, this hurts. But it itself doesn't feel pain. So if you're going to have brain surgery, they don't put you to sleep because you're not going to feel it. And if they keep you awake, they can keep you talking. And so they know that if you stop talking or if you start talking funny, that they're doing something wrong. So they keep you awake for it. But so you say, well, but wait a minute, because I have headaches. So if it's not my brain hurting, what am I actually feeling? What is all that pain that I have? Well, a lot of times your headaches are vascular spasms. The blood vessels are spasming. I am almost positive that you have heard somewhere in your life that if you don't receive oxygen, if you don't have blood flow, if you're not breathing, heart's not beating for 4 minutes, you have irreversible brain damage. So that has always been in all the textbooks and all of that. And there was a child who was sledding or skiing, I think he was sledding, and he went into a lake. He broke through the ice, and he was underwater for half an hour. So obviously he was not breathing underwater for half an hour, so he'd been dead. And so they pulled him out, and they said, well, even if we were able to revive him, then his brain would have been destroyed, and, but they went ahead and they revived him, and his brain was just fine. So you need to put like a little asterisk here and say, but if you are cold, super cold, then you don't get that irreversible brain damage nearly as quickly. But if you're at room temperature, then you're going to have brain damage if you stop breathing or stop blood flow for 4 minutes. On the other slide, we talked about how cerebrospinal fluid is different from plasma. So it doesn't have as much calcium and it doesn't have as much of these other things. So how do you keep the cerebrospinal fluid having its composition separate from the blood, which has plasma? So that's where you talk about your blood-brain barrier. And it is selectively permeable. So it likes water to come through into the cerebrospinal fluid. You need that. And it really wants glucose because the brain has to have glucose. It can't make its own enough to keep it going. And you need lipid-soluble substances. You need oxygen, carbon dioxide. Now, you don't need alcohol, but it can easily go through. So alcohol goes through the blood-brain barrier, caffeine goes through the blood-brain barrier, and so does nicotine and things that put you to sleep, anesthetics. It is slightly permeable to sodium and chlorine, but now remember, we don't want it to be too permeable because we want more sodium and more chlorine in the cerebrospinal fluid than we have in the plasma. So you have to be careful because permeability goes both ways. And of course, waste products, urea, creatinine. So how could blood get into the brain? Well, it gets in through capillaries. So you have to guard the capillaries to make sure that that plasma doesn't leak over into the cerebrospinal fluid, the CSF. When we learn about the blood system, you're going to learn that capillaries are supposed to be leaky. They're supposed to allow fluids to leak out into the tissues. So that's how the nourishment gets out to the different cells that the capillaries are running past. But in the case of the blood-brain barrier, you don't want the capillaries to be leaky. So you have the astrocytes, and that's the most plentiful of the neuroglia. They have projections coming off of their cell membrane, and they touch the capillaries, and they seal off any holes that might be between the squamous epithelial cells of the capillaries. So it forms tight junctions between the endothelial cells or the squamous epithelial cells. So it completely seals off the gaps. So anything that gets out of the blood and goes over into the cerebrospinal fluid has to go through the cells, not between the cells. So you can choose what you want to come into the CSF and what you want to stay in the plasma. I remember a number of decades ago when I was doing Alzheimer's research that they found high levels of aluminum in the brains of people who have Alzheimer's. and they thought, aha, we have maybe found a cause for Alzheimer's. But with more study and more looking at it, because the brain is breaking down, because the cells are dying, you are unable to keep the blood-brain barrier intact. And so you don't really think about all the aluminum you're exposed to. I mean, you slather aluminum on under your arms every time you put on antiperspirant. So we're exposed to a lot of it. You drink a lot of your pop, whatever carbonated beverage you have, out of aluminum cans. So we get a lot of aluminum, but the brain is protected by this blood-brain barrier. But people who have Alzheimer's, it breaks down. So the consensus was it's not the aluminum that's causing the Alzheimer, but the fact that the person has Alzheimer is allowing aluminum into the brain. On many levels, the blood-brain barrier is very beneficial, for maintaining homeostasis and keeping things out of the brain that you don't want in the brain. But unfortunately, there's some things you would like to get into the brain, antibiotics to cure meningitis, or cancer drugs on the off chance that the cancer is spread to your brain. But the blood-brain barrier will block those. Another one is people who have Parkinson's. They lack dopamine. They can't make enough dopamine to function correctly. And if you could just get the dopamine to go across the blood-brain barrier, then you could just give them dopamine. But if you watch the movie Awakenings that stars Robin Williams, he said, well, why don't we give huge doses, really huge, scary big doses of dopamine and see if it'll go across the blood-brain barrier and get in. And if you give really large doses, then you can get it across the blood-brain barrier. But it's not practical, and you develop a tolerance to it very quickly. So, but if we could figure out a way to transport things across the blood-brain barrier, that would be pretty neat. When we continue this with part 2 of chapter 14, we'll start off talking about strokes.